Just going to give everybody a few more minutes to join. Hello, everybody. Welcome. We're giving everybody just another minute to join for those that arrived a little late. All right, looks like we have enough people. So I'm going to go ahead and start. I wanted to say hello and thank you for joining us. Today, Business Development Director Dan O'Connor will introduce our speakers who will present on developing a PBPK modeling approach for evaluating drug product performance in infants based on adult data. A few housekeeping notes before we begin. We take your privacy rights seriously. By attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. And before I pass things off, I have a poll question for all of you. Are you currently using a modeling and simulation software package for your research? And I'll give everybody a few more seconds on this. So it looks like most of you are. This is good. And I'll now pass things off to Dan.
Good day, and thank you, Jasmine. I'll turn my camera on just for a moment to say hello to everybody, and I'll turn it off so that you don't see me reading. My name is Dan O'Connor, and I'm very happy to be able to welcome you to the first of a new series of webinars that we're hosting as part of our celebration of our 25th anniversary. Simulations Plus has a long history, I think most of you know, supporting academic research. And we're excited to be able to highlight the modeling and simulation work that's being conducted by academic researchers worldwide. In addition to today's presentation, in the coming months, we have 2 additional webinars that we would ask you to consider. They're scheduled, the second is with the University of Florida on November 16th, followed on December 15th with a presentation from the University Federal São Paulo, Brazil. Today's presentation will be tag teamed by Drs. Marina Stepalova and Maria Bertazzoni. And I'll just give you a little bit of their background. Marina is a pharmacist who graduated from Frankfurt am Main, Germany. She was an early stage researcher in the Pearl Consortium, conducted her PhD studies with the Department of Pharmacy and National Custodian, University of Athens, under the supervision of Professor Maria Bertoni, who is also presenting today. Marina's PhD work focused on developing in vivo and in silico biopharmaceutic tools for the evaluation of pediatric drug products. So perfectly illustrative of what we're presenting today. She's co-author of many research papers and review articles as well. Our first presenter today will be Maria Bertazzoni. Maria is an assistant professor in pharmaceutical technology and biopharmaceutics, Department of Pharmacy with the University of Athens in Greece. She received her baccalaureate in chemistry in 1994, her master's of analytical chemistry and her PhD, all from NKUA. In 2016, received her Master of Science in Medical and Pharmaceutical Statistics and is focused in on the physicochemical characterization of intraluminal environmental, oral drug absorption, and special interest in special populations. Currently, she's supervising 4 PhD theses, so incredibly that she has time for us today. Thank you. And without further ado, I'd like to introduce Dr. Verdezzoni.
Thank you for the kind introduction for both of us. We're both happy to be here with you and within the GastroPlus user group. So the title of our presentation will be Developing a PBPK Modeling Approach for Evaluating Drug Product Performance in Infants based on adult data. The development of pediatric formulations and the extension of licensed drug use to the pediatric populations has become an essential part of the drug development process in order to ensure appropriate dosing, efficacy, and safety for all target pediatric age groups. However, ethical concerns and the recruitment issues can limit clinical testing in these different age groups more than in adults. In addition, differences in gastrointestinal physiology across pediatric subpopulations and differences in meals administered to evaluate the impact of prandial conditions can increase data variability and can also decrease their usefulness. In this presentation, we will discuss the current status of the evaluation of drug product performance in infants, and we will focus on the usefulness of studies in healthy adults. Initially, we will examine if differences in dosing conditions between oral drug absorption studies in adults and oral drug absorption administration to infants impact PBPK parameters. And then, based on data collected from clinical studies in adults, Marina will describe and show you the evaluation of a PBPK modeling approach for extrapolating drug exposure in adults to infants under different dosing conditions. So in recent years, there has been an interest in investigating whether food effect data collected in adults are useful for pediatric products. Based on the draft guidance issued by FDA 2 years ago, when a new pediatric formulation is developed, the sponsor should conduct a new food effect study with a pediatric formulation in adults and then extrapolate the results to the pediatric population. A concern in regard to this relates to the design of food effect studies in adults. So the questions that comes is the effect observed after a high-caloric, high-fat meal similar to the effect of infant food in adults? And are dosing conditions similar? To date, bioavailability and food effect studies for orally administered pediatric products are performed in adult volunteers according to regulatory guidelines, with the application of suggested fasted conditions. That means the formulation is administered with a glass of water. Now, in order to achieve fed conditions, FDA suggests the consumption of a high-calorie, high-fat solid liquid meal with approximately 1,000 kilocalories and approximately 60% fats 30 minutes before the administration of a drug product. By employing a high-calorie, high-fat meal, food effect studies in adults aim at detecting the maximum effect on GI physiology and the maximum effect on bioavailability. But is the effect observed in adults after the consumption of a high-calorie, high-fat meal similar to the effect of infant food in infants? We try to review food effect investigations regarding drug exposure in infants. To date, 9 data sets from food effect studies for 7 different drugs in infants and young children have been published. This means the range of from 2 months to 2 years old. Within these investigations, the tested product was an antibiotic suspension and the tests were performed on a predominantly crossover basis. The fasted state was defined as a period of 2 hours before and after drug dosing, during which no food or milk-based feed was allowed, and to induce the fed state, milk or infant formula were co-administered with the product. This means feed was administered immediately after drug administration or simultaneously with the drug. We summarized the outcome of these investigations regarding drug exposure, and we compared them with the food effects reported in adults from the same antibiotics. These adult investigations were performed on a crossover basis using an immediate release product, and the test product was administered after overnight fasting to study drug performance under fasted conditions, and 0 to 1 hour after a solid liquid meal to study the fed state. The outcomes of these food effect investigations for the 7 antibiotics tested are summarized in this slide. As we can see in this slide, the food effects for pediatrics and adults are not always the same. So, as we can see, only erythromycin ethylsuccinate seems to have comparable food impact in infants and in adults. This data, however, should be interpreted with caution, as the majority of this data have been collected more than 40 years ago. And if we go through the composition of infant meals, we can see the caloric density values of commercially available infant formula compared with the human breast milk. And as we can see, they are similar, given that infant formula development is based on the properties of human breast milk. These two types of infant formula exhibit similar macronutrient composition, as we can see in this table, and their caloric content can be characterized as high carbohydrate and relative high fat but low protein. And here is the composition with this comparison with the standard high calorie, high fat meal as proposed by the regulatory agencies. As we can notice, the caloric breakdown of the standard adult meal differs from the composition of infant formula and the composition also of infant meal. So the design of food effect studies in adults appear critical when food effect data on oral drug absorption in adults are to be extrapolated to pediatric populations. The recent guideline by FDA on how to conduct food effect studies for newly developed pediatric formulation suggests that the food effect for pediatric formulations could be evaluated in adults using foods and quantities of food that are commonly consumed with drugs in pediatric population, with then an extrapolation of the results to the pediatric population. However, with the draft guideline, the appropriate meals, the quantities, but also the dosing conditions are not specified. To this aim, we designed an exploratory food effect study in adults in order to investigate the usefulness of studies in healthy adults and to understand if differences in dosing conditions between oral drug absorption studies in adults and oral drug administration to infants impact PK parameters. In our case, we selected 2 model drugs based on their luminal stability and high intestinal permeability. So one class BCS class 1 and one BCS class 2 API paracetamol for class 1, which has salt solubility, and ibuprofen, a low solubility and weak acid with pK 4.5 were selected and were co-administered using commercially available pediatric suspensions. That means the variation of dosing should impact primarily gastric emptying in the case of paracetamol, or gastric emptying and perhaps the solution for ibuprofen. This study was a single dose randomized crossover 3-phase comparative oral bioavailability study with a washout period of 1 week. A total of 8 healthy adult volunteers completed all 3 study phases. Now let's see these phases. In phase 1, the formulations were administered with water So the total fluid volume of the administered formulations and the water was 250 ml in the following manner within 2 minutes. So the formulations were administered sequentially without time gaps in between, and time zero was just after the completion of the first minute. In phase 2, the formulations were administered as described for phase 1, but 30 minutes after the initiation of the ingestion of the FDA meal with a total caloric content of about 1,000 kilocalories. For phase 3, the infant formula was selected as an age representative meal in the pediatric subpopulations below the age of 2 years old, based on its frequent use. The volume of infant formula in the present study was 800 ml, that means 520 kilocalories, and was based on the recommended infant formula volume for infants, scaled up by a body surface area factor for adults versus infants. To simulate dosing condition in infants during feeding, the total volume of infant formula was split in 2 portions, and the 400 ml were consumed at a constant rate over 8 minutes, and then 2 ml of Nurofen and 21 ml of Panadol were administered over 2 minutes, and upon completion, time zero was set, and then the administration of Nurofen and Panadol was continued over a period of 2 minutes, after which the second portion of infant formula was consumed at a constant rate over 8 minutes. The formulations and infant formula were again administered sequentially without time gaps in between. And let's see the results. In the figures with white circles are presenting plasma concentrations under fasted conditions, with black squares the fed conditions after the reference meal, and with purple triangles the infant fed conditions. This means the co-administration of the drug products with infant formula. Paracetamol total exposure, that means Cmax, you see, Cmax and Tmax values were not significantly influenced by the various dosing conditions applied in this study, as we can see in this table. Based on partial AUCs calculated based on the Tmax observed in the fasted state conditions, early exposure under fasted conditions and fed conditions demonstrated no significant difference. But under infant fed conditions, early absorption of paracetamol was significantly slower than the fasted state, despite the lower total caloric content of infant formula compared with the meal used to induce fed conditions. Same observations we have when we calculate the partial AUC based on the Tmax observed under fed conditions, and similar results when the Tmax of infant fed formula conditions is used as a cutoff point for the calculation of AUC. So early absorption of paracetamol was significantly slower under infant fed conditions than in the fasted conditions. As for paracetamol also, we have similar results for ibuprofen. Total exposure was not statistically different among the 3 study phases, but peak exposure under infant fed condition was significantly greater than the observed under fed conditions. Dosing under infant fed conditions significantly reduced early exposure compared to the fasted conditions during the first 45 hours after drug administration, and under infant fed conditions, partial AUC values were significantly lower compared to the fasted conditions. So under infant fed conditions, all partial AUC values were significantly lower compared to the fasted conditions. Summarizing the key results from this exploratory clinical study in adults, we can conclude that no significant effects regarding total exposure were notified, but there was a decrease in early exposure under infant formula-fed conditions compared to the fasted conditions. Differences could be attributed to the gastric mixing events and the gastric emptying of drug with or without the administration of the meal, and assuming that the food type rather than age is the main determinant for gastric emptying, data from this study indicate a subsequent delay in paracetamol or ibuprofen absorption, and probably a delayed induction of pharmacodynamic effects when a suspension is administered during feed with breast milk or infant formula in infants. And based on this study, Marina will describe you how we develop and evaluate the PBPK modeling approach in order to extrapolate drug exposure in adults to pediatrics under these different dosing conditions. Marina, the stage is yours.
Thank you very much, and welcome everybody. As Professor Verdoni just said, I'm going to take over for the next a couple of slides and describe and evaluate a PBPK modeling approach for extrapolating drug exposure in adults to pediatrics under the different dosing conditions we just described. So starting with first things, I can't change the slide. Could you please go to the next slide?
Yes.
Perfect. Starting with the in silico tools in pediatrics, one of the most used ones is the physiologically based pharmacokinetic or PBPK modeling. As probably most of us know, physiologically based modeling strive to predict drug performance a priori by simultaneously considering drug-dependent properties, physiology and anatomy properties or system parameters, and take into account clinical trial design. Having said this, this allows scaling for different ages and different species, which is extremely important when we model data to pediatrics. Here in this figure, we show you a review article that research we performed, and it is published in the review article. We can see the basis for PBPK models developed for pediatric populations, and what we can see here is that more than 80% of those models were based on adult data. Several of them refined the models, including pediatric populations or animal studies, but the biggest portion was based on adult studies. Now we would like to show you the modeling workflow we followed. And as a first step, we started with our adult disposition model. For this purpose, we had our system parameters, our drug physchem properties, our biodependent properties, and included some ADME data. This development was evaluated and refined using IV data in adults, and using an external set of PK studies in adults, we verified our model. In the next step, we entered our drug formulation, which was a solution. We entered our drug permeability and included GI physiology at this step, and verified the first part of our oral model. In the next step, we added our pediatric suspension. We investigated different dosing conditions as the ones mentioned already from the first study. And if needed, we performed some GI physiology adjustments. As we said, following the verification of our oral absorption model in adults, we continued to our pediatric model. At this stage, we included age-specific changes regarding physiological parameters such as drug binding, so plasma binding or blood to plasma ratio. we included we performed clearance scaling, and finally we extrapolated the dosing conditions mentioned in the previous studies to the pediatric population. This model or the performance was evaluated using PK parameters, using pharmacokinetic, so the profiles in pediatric populations that were reported in literature. We performed all this work using GastroPlus version 9.7, and the PK analysis was performed using PK Plus. Having said this, our modeling involves quite a rich clinical PK data set, part of which came from literature, of course. We had as a total 23 pharmacokinetic data sets in adults and pediatric patients. 17 data sets were used from adults, 12 from those were coming from intravenous administration, and 5 data sets were from oral drug administration. In 2 studies, solution was studied under fasted conditions. This one we modeled to confirm our permeability parameters. And then for the adult model, we used our pediatric suspension under different dosing conditions, which was presented in the first part. And for the pediatric studies, so the studies in pediatric patients, we had as a total 6 data sets. 4 of them followed intravenous administration to confirm our disposition and clearance scaling for pediatric patients, and 2 data sets followed oral administration in infants or infants and young children. One of these studies investigated paracetamol suspension administration, and here our population was only infants, and the other study followed administration of paracetamol elixir, and here we had a bit broader population. Having this said, we will start directly from the adult absorption model, where we model the suspension given as 100 milligrams of paracetamol, and we created our mean population representative. We performed here single simulations. We followed the study mean physiology, so we created a 78 kilogram, 28 years old virtual physiology. In the graphs below, we can see from left to right the different dosing conditions and of the observed data. So starting with the fasted conditions, reference meal fed conditions, and infant formula fed conditions. With the squares, we can see the mean observed data, and the gray lines represent the observed individual data. We started first by applying the default settings within GastroPlus. Here we started with the default setting for the fasted physiology, as shown in the dotted line. Following, we utilized the new function of GastroPlus to calculate a user-defined meal. So here we could enter our meal calories. So for example, for the reference meal, we entered our 1,000 kilocalories, and additionally the percentage fat in the meal, which were 60%. Based on that, the software adjusted the gastric transit time and the bile salt concentration within this physiology. And the same way of work was followed for the infant formula fed conditions, where we entered our 520 kilocalories for the whole meal and 43% fat within the meal. Again, we had here an adjustment of the gastric transit time and the bile salt concentrations. Having applied those settings, we can see that, again, with the dotted lines, we did get the direction of absorption delay. However, some refinement was needed in both cases. Having said so, we adjusted the gastric transit time for the fasted state to 0.75 hours to match the delay observed in vivo, shown with a solid line. Then for the reference meal fed state conditions, we adjusted the gastric transit time to 1.5 hours. Here the gastric emptying followed first order kinetics. And in the last condition, so under infant formula fed conditions, we applied zero order gastric emptying as known for homogeneous liquids emptied from the stomach, and the gastric transit time, which in this case represents the total gastric transit time, was adjusted to 4.5 hours. As you can see, both from our evaluation parameters and visual checks, we had here successful simulations of the 3 dosing conditions, and we proceeded to scaling those conditions to pediatrics by keeping the same gastric emptying order for each condition and by scaling the gastric transit times for each condition. We're going now over to our first case study or first infant study data set we had. This is the Hopkins et al. Study published in 1990. The population included 5 infants with the mean age of 4 months, mean body weight 4.3 kilograms, and the dose administered, the mean dose administered was 19.6 milligrams per kilogram. Based on that, we created a virtual physiology for our mean population representative, which was again 4 months, 4.3 kilogram male, corresponding to the study. And this is how it looked like in GastroPlus. So here we entered our the age and kilograms, and our virtual physiology was created. To come to the scaling of our gastric transit time or our conditions for the two fed conditions, starting from our reference meal, which is a solid liquid meal, we kept the first order gastric emptying and used the caloric content used in the adult study and the adjusted gastric transit time to calculate the pediatric volume, the respective pediatric calories. The same was performed also for the infant formula, which had 520 kilocalories, and the adjusted gastric transit time from the model was 4.5. The adjustment was based on the meal calories, on the meal caloric content of an infant meal, and was normalized with a paracetamol gastric transit time from adults, and was normalized with the caloric content of the adult meal. What this means for the 4-month-old population representative is that we had meal calories of 140 kilocalories that we scaled based on our adult model and adult data. And this gave us an adjusted gastric transit time for the fed state of 0.21 hours. And for infant formula conditions, this gave us, this resulted in 1.21 hours. Starting with our simulations for this study and applying firstly our fasted conditions. As we said, we kept the gastric emptying order observed in adults and the gastric transit time. We did not change this because we had fasted conditions. And in this figure, we can see again with the purple line our simulated concentrations, and with the circles and gray lines are observed data in our observed clinical data in pediatrics. The profile shape is in line with the observed data. The simulated profile shape is in line with the simulated data. And if we look on the bottom of the slide, here we present the fold difference for the different PK parameters. So AUC Cmax, Tmax, and AUC until the Tmax, so the partial AUC of early absorption of the oral absorption phase. And we can see with the purple, this is the line of unity, and our predictions are quite close to our simulations are quite close to unity. Looking at our average fault error, again, this indicates a good simulation. We're again close to unity, and looking over to the absolute average fault error, or AFE, here we are close to 1. Our simulations are close to 1, which means we have adequate precision of the simulation. Coming now to the second condition we model, this is the reference meal fed conditions. Here, keeping the gastric emptying order from the adult study and using the adjusted gastric transit time of 0.21 hours, we can see that we overpredict the early phase, which is also reflected with our squares in the tables below. So we can see our Cmax is overpredicted, Tmax is underpredicted, and the partial AUC is on the borderline of a twofold prediction error. Again, average fault error and the AFE were not really affected. Looking into our third conditions, which is the infant formula, fed conditions. Here we applied zero order gastric emptying and the scaled gastric transit time from adults. And as indicated from the purple line, we can see the profile shape is in line with the observed data. And if we look at our parameters for the model, so our evaluation parameters, we can see with the triangles that were for the fold differences were always quite close to unity. The same applies for the AFE and for the AAFE values. Coming now to our second study data set, which is the Walson et al. Study. Here, the data set was from 12 infants with a mean age of 10 months, with a mean body weight of 8.6 kilograms, and the mean dose administered here were 12 milligram per kg. We, again, following the same way, we created our mean population representative physiology, and this was a 10-month-old, approximately 9 kilogram male. And again, using the paraphysiology module, we created the virtual physiology, the respective virtual physiology with the tissues and organs. Again, as for the first study, this time using the meal calories for a 10-month-old population representative, those are 170 kilocals. We calculated adjusted GTT for a solid liquid meal, meaning 0.26 hours, or for infant formula meal or homogeneous liquid meal, around 1.5 hours. Following the same routine as for the first data set, here the observed data is presented with the black circles, and again our simulation is presented with the bold line. We can see that the fasted conditions were well in line with the observations. Same goes for the fold difference. Although the Tmax is slightly overpredicted, this is probably due to the nature of the data we had, and average fault error and AFE are well in line. So the simulation was considered adequate. Applying the reference meal fed conditions, here again using first order gastric emptying and the adjusted gastric transit time, we can see that the profile shape is rather in line is in line with the observations. And finally, looking at our evaluation criteria, we are within twofold error. Lastly, applying our infant formula fed conditions, or using gastric emptying of 0 order and the adjusted gastric transit time, we can see that our model overestimated the delay observed. So we can see we are mismatching the initial part, the absorption part. This is also reflected in our evaluation criteria here with the triangles again. We can see that the Tmax is over predicted and the AUC and the partial AUC is rather under predicted. So the infant formula appeared less useful in this case. Now I will guide you to our concluding remarks, and starting with the first conclusion is that even for drugs with non-problematic absorption administered in simple dosage forms, food effects in infants might not be adequately evaluated if the protocol suggested by regulatory agencies is applied in adults. Then we proposed and evaluated a methodology where we administered pediatric formulations under 3 different dosing conditions in healthy adults. Then use this dedicated relative bioavailability study to inform or as a basis for our PBPK model and GastroPlus. And then we extrapolated or scaled this model to our pediatric population. This proposed methodology to predict pediatric formulation performance deserves further evaluation using high-quality clinical data, both in adults and infants, but from our study, this appeared quite a promising methodology to follow. Having said this, we would like to acknowledge all our co-workers from the National Encapistrian University of Athens and from the Pearl Consortium who collaborated with us and were always very helpful. And special thank you to Professor Reppes, who spent quite some hours with us in the hospital as well. And with this, we would like to open the floor to any questions, and thank you for joining us and for tuning in to the presentation.
Thank you, Marina. Before we jump to questions, I do have one more poll to follow up on our previous one. And this asks, which software platform are you currently using if you are using one? And you can select more than one answer here. I'll give you guys a few more seconds here. All right, and it looks like most of you are using GastroPlus. And now we can kick off the questions. I'm going to go ahead and pass it off to our Vice President of Regulatory Affairs, Sandra. I think you're on mute, Sandra, if you're talking.
Yes.
There you go.
Just want to show my camera for a second. Hello, everyone. It's a pleasure to be here. Maria and Marina, very nice presentation. I think that you have created a topic for several discussions and questions that have been raised. Let me just go through the questions and see. So, Maria or Marina, the question is, how delayed peak plasma time is explained in the pediatric fed state?
Thank you, Sandra. I will take this question. It's a very interesting question, and it's very interesting to know how gastric transit time changes in fasted and the fed state, and how the type of milk can have an impact on this gastric emptying. So the main difference is that in the adult study, we have a solid liquid meal, and after 30 minutes, we administer the API or the drug product with a glass of water. And as stated in the references and in the literature, and mainly in works made by Werner Weitz's group in Greifswald, we have this stomach road, as known as Magenstrasse. So the product disintegrates, in our case, the suspension, may empty with the water administered. So that's why we notice more or less similar performance in the profiles between fasted and fed adult meal conditions. On the other hand, when we have the infant meal formula, we have an homogeneous meal, and we'll co-administer under pediatric conditions the drug product, as usually we're doing at home with children, but refuses generally to take their API in order to convince them. We try usually to give them with infant formula. That's why we try to simulate these conditions, not only because the regulatory agencies try to push to this way, but also because it's their life. So in real life, when we have the infant formula, we co-administer your drug product. The drug product can disperse and dissolve in this homogeneous meal and can empty together with zero order ending as it's homogeneous. This is why we can say that we have these differences between the two meals. I don't know if Marina would like to add something on this.
Yes, exactly. So I think you covered it nicely. Exactly. So the main difference is here that we have different textures between the two meals, and that our formulation could mix rather well with the homogeneous infant formula, as opposed to the solid liquid FDA meal, where probably portions of the formulation and the drug were able to empty from the stomach, independent of the main meal chyme.
Great. Well, there is several questions here. Let me raise another question. How do you change gastric emptying order? Maria? Do you wanna?
Yes, it's very interesting, and it's nice that we can do this within GastroPlus. Maybe Marina can respond to these technical questions, since she's more experienced and she's did this work. Marina?
Yeah, exactly. So there is this option within GastroPlus where you can just basically tick the box above the physiology to change the gastric emptying kinetics. However, here we need to be cautious about the gastric transit time, as for a first order emptying process, the gastric transit time depicts the mean gastric transit time, while for the zero order, this depicts the total transit time. But there is a box on the platform in the physiology tab, in the GI physiology tab.
Thank you very much, Marina. Another question. In terms of partial area under the curve, how much variations exist between clinical studies, especially fed conditions? Maria or Marina?
It's a very general question, and this relates also to the way of administration of the product, and also in our case, for early exposure, as it depends on the emptying also of the meal, we can have a high variability, and that's why we performed the ANOVA test in order to find and identify if we had the significant differences or not, just to take into account also this variability in our clinical study.
Great. Let me see other questions here. Could you please comment on the modeling to predict pediatric absorption for BCS classes 2 and 4? I don't know if that question is...
It's a very nice question, interesting question, but as Marina pointed out in the concluding remarks of this presentation, is very interesting, and we need the evaluation of high quality clinical data from both adults and infants in order to be able to make good predictions and good simulations. So for the moment, in regards to the pediatric products and knowledge, we have run this clinical study for BCS class 1 and 2 products. We used suspension because it's all that are available in pediatrics. And it would be very nice to have more data and more points in order to be able to evaluate the simulations. So it's not easy to make conclusions, and without having in mind that the lack of quality clinical data is the limiting step in order to propose something good.
Mm-hmm. Thank you, Maria. Let me see several questions here. Another one. Is there any way to fit the individual data rather than just the mean PK profiles in GastroPlus?
Maybe I can take that one.
Sure.
Yeah, perfect. So yes, we started off with this approach. And as you might have seen probably from the profiles in adults in all three different dosing conditions, we observed some erratic behavior, erratic absorption behavior, meaning we had both double peaks or shoulders. And this one you can model actually quite nicely within GastroPlus if you're sure that that those peaks are gastric emptying driven. You can use the multiple physiology mode, and you can basically introduce different gastric emptying events. Through this, we were able to replicate the observed data, both for the fasted state and also for the fed conditions. However, at this point, it was very interesting to understand our clinical data in adults, but for the extrapolation to pediatrics, this was of, let's say, limited value to understand the discontinuous gastric emptying behavior, as we do not know it as such for the pediatric population. But you can surely do it for adults.
Mm-hmm. Maria, we are good with that. Yeah, thank you.
Yes, for sure.
All right, so there is another question here. In terms of partial area under the curve, how much variation exists between clinical studies, especially fed conditions? I think.
Yes, if we pass through this, yes, more or less.
Sorry, what did you...
It is almost the same as the previous one, yes.
Right. And I don't think we have any other questions being posted right now. Is anything else you wanted to add, Maria?
No, I would like to say that we were happy to work with GastroPlus 9.7 because, as Marina pointed out, it gave us the opportunity to use this user-defined meal. Otherwise, with the previous version, I don't know if this would be possible for us, and it was a nice opportunity to work with this. So every improvement is more than welcome.
Sure. It says here, can we share the slides deck with the attendees? I think, what do you think, Maria? Can we share the slides with them?
Yes, for sure. You can share the PDF sent you with the attendees. And if there are any comments, please feel free to contact directly us.
Great.
Awesome, thank you. So I will doing our final remarks. I just wanted to say thank you all for joining us today. We invite our audience to learn more about GastroPlus on our website at simulations-plus.com. This webinar has been recorded for playback, so we will have the slides and the video available on our website and YouTube channel. Was there anything else we wanted to touch on, Deanne or Sandra?
No, I just want to again thank you everyone for attending this webinar. And don't forget that, as Dan mentioned at the beginning, that we are going to have other sessions coming up soon. So stay tuned. Thank you again.
Thank you, guys.
Bye.
Thank you again, Maria and Marina. Thank you for your
Thanks,
participation.
everyone.
Bye-bye.
Bye-bye.